When an EMS crew rushes a patient through the bay doors in total ventilatory collapse, they are watching a highly specific neural circuit fail in real time. We typically associate somatic control with moving our arms and legs, but this exact same pathway governs the vital voluntary breathing muscles, specifically the diaphragm and the intercostals. When this circuit drops, the patient stops breathing. At its core, the somatic nervous system is built on a very specific formula. It combines conscious, physical sensation with voluntary skeletal muscle control. The architecture here is distinct. While the autonomic nervous system uses a two-neuron chain to reach its target, the somatic system relies on the one-neuron rule. A single, uninterrupted motor neuron extends all the way from the central nervous system directly to the muscle fiber. This single, unbroken physical wire makes somatic reflexes and voluntary movements incredibly fast, but it creates a massive biological vulnerability. With no backup circuits, if that single wire is cut or blocked, the muscle is instantly paralyzed. Every conscious movement begins with a continuous feed of environmental data. This is the afferent division of the somatic system, which carries sensory input from the periphery up to the central nervous system. Different receptors capture entirely distinct data streams. In the skin, Meissner corpuscles and Merkel discs map touch and pressure. Pacinian corpuscles detect vibration. Meanwhile, bare, free nerve endings transmit raw pain and temperature data. To coordinate movement, the brain requires proprioception, knowing exactly where a limb is positioned in space. This spatial awareness relies on specialized proprioceptors. Muscle spindles physically measure stretch within the muscle belly, while Golgi tendon organs gauge tension pulling on the tendons. This dense stream of afferent data feeds upward, allowing the brain to process joint positions, track muscle tension, and execute high-speed, real-time movement corrections. Without this high-fidelity sensory feedback, the brain is flying blind. Precise, coordinated motor output is impossible without knowing the exact starting position of the body. The efferent side, the motor output, begins in the primary motor cortex, located along the precentral gyrus of the frontal lobe. This origin point is the upper motor neuron, or UMN. It fires the moment you make a conscious decision, whether that is deciding to walk across a room or reaching out to grab a laryngoscope. Once triggered, the electrical signal travels down the central nervous system's main descending highway, the corticospinal tract. As the signal hits the medulla in the brainstem, it encounters a critical anatomical intersection known as the pyramidal decussation. Here, the vast majority of motor fibers physically cross over to the opposite side of the spinal cord. Because of this decussation, the left side of the brain controls the right side of the body. Clinically, this explains why a stroke or brain bleed in the left hemisphere presents as contralateral weakness, paralysis on the right side of the patient. The surviving signal continues its descent down the spinal cord until it reaches the ventral horn. Here, the upper motor neuron synapses with the lower motor neuron. The lower motor neuron then exits the spinal cord. It travels out through the ventral root, merges into a peripheral nerve, and acts as the final physical wire heading straight for the skeletal muscle. The ventral horn serves as the vital relay station. It is the exact anatomical location where a conscious central brain command is handed off to the body's peripheral wiring. Sometimes, waiting for a signal to travel from the cortex, down the tract, and back out again takes entirely too long to prevent physical tissue damage. To solve this, the somatic system uses reflex arcs. These are specialized pathways designed to maximize speed by intentionally bypassing the conscious cortex during the initial reaction. The most direct version is the monosynaptic reflex, commonly tested as the patellar knee jerk. A tap on the tendon suddenly stretches the muscle spindle. The sensory neuron fires directly into the dorsal horn of the spinal cord, synapses directly onto the alpha motor neuron, and the quadriceps instantly contracts to kick the leg. The withdrawal reflex operates slightly differently. If you touch a piece of hot metal, pain receptors fire an urgent warning signal to the spinal cord. In this case, an interneuron in the spinal cord intercepts the data. It rapidly splits the signal. One impulse forces the flexor muscles to contract and pull your hand away, while the simultaneous inhibitory impulse forces the opposing extensor muscles to relax. These reflexes prove the spinal cord is an active processing center. It functions as a localized survival hub, engineered to prioritize immediate physical safety over conscious cortical thought. When we follow the lower motor neuron to its absolute endpoint, we reach the effector organ. For the somatic system, this is strictly restricted to skeletal muscle. This intersection between nerve terminal and muscle fiber is the neuromuscular junction, or NMJ. In clinical medicine, it is the single most important somatic receptor site. The physiological rule here is absolute. To cross the microscopic gap between the nerve and the muscle, the somatic system relies entirely on one neurotransmitter, acetylcholine. The acetylcholine diffuses across the gap and binds to a highly specific target on the muscle side, the nicotinic NM receptor, with the NM standing for neuromuscular. This receptor functions as a ligand-gated ion channel. When the acetylcholine binds, the channel physically unlocks. Sodium rushes into the cell, potassium rushes out, and the resulting shift in electrical charge creates cellular depolarization. This localized end plate potential triggers an action potential across the muscle fiber. Calcium is released from the sarcoplasmic reticulum. The actin and myosin proteins form a crossbridge, and the muscle mechanically contracts. Every conscious human movement, from speaking to swallowing to expanding the diaphragm, depends entirely on this microscopic chemical exchange. That reliance makes the NMJ the ultimate bottleneck for neurological disease. When this system fails, the physical symptoms presented by the patient map directly to the exact anatomical location of the lesion. Upper motor neuron lesions occur entirely within the central nervous system. These are caused by strokes, brain bleeds, spinal cord injuries, or diseases like multiple sclerosis. The physical result of a UMN lesion seems counterintuitive. Because the intact lower motor neuron is suddenly cut off from the dampening inhibitory signals usually sent by the cortex, it begins firing wildly. The clinical presentation is spastic paralysis, hyperreflexia, and intensely increased muscle tone. Lower motor neuron lesions present the exact opposite picture. These are caused by peripheral nerve injuries, spinal root compression, or ALS. In these cases, the physical wire connecting the spinal cord to the muscle is completely severed or destroyed. The poliovirus is a historical masterclass in lower motor neuron destruction. It specifically targeted the anterior horn cells, where these lower pathways begin, wiping out the somatic connection to the diaphragm and causing widespread ventilatory collapse. Because the muscle receives absolutely no signal, an LMN lesion results in flaccid paralysis, absent reflexes, rapid muscle atrophy, and visible twitching beneath the skin, known as fasciculations. As a clinician, you can instantly localize where the nervous system is broken. If a paralyzed limb is spastic and rigid, the damage is central. If it is entirely flaccid, the peripheral wire is cut. The third major somatic failure point exists at the neuromuscular junction itself. Diseases here leave the nerve and the muscle intact, but destroy the chemical bridge between them, as seen in myasthenia gravis. In myasthenia gravis, the patient's own antibodies attack and destroy the NM receptors. To secure a compromised airway, clinicians administer chemical paralytics like succinylcholine or rocuronium. These drugs are designed to forcibly block the NM receptor. By occupying the target sites, they intentionally shut off the entire somatic motor pathway, paralyzing the diaphragm and skeletal muscles long enough for a breathing tube to be inserted. Every intervention relies on understanding the master flow. The cortex signals the corticospinal tract. The signal hits the ventral horn, travels down the peripheral nerve, releases acetylcholine, binds to the nicotinic NM receptor, and triggers skeletal muscle contraction. It is a single, continuous biological circuit. Mastering its anatomy is precisely what allows advanced clinicians to intercept neurological failure before it becomes total respiratory collapse.